right, so today I'm going to tell you about the impact of vitamin B12 on amyloid-beta induced proteotoxicity. And this was actually a project that we stumbled upon accidentally when I was an INBRI pilot investigator. And we were studying genetic factors that could potentially impact amyloid-beta induced proteotoxicity. And in the process, we discovered that diet was having a role. And I work with the model organism C. elegans. And just so you know, this image on my title slide, this is actually a worm, and it's false colored, but it's expressing a sensor that detects vitamin B12. So there are currently 6 million Americans that have Alzheimer's disease. This is the most common cause of dementia. It's predicted that by 2050, over 100 million people in the world will be afflicted with this horrible disease. And given the great emotional, social, financial impact that Alzheimer's disease has on both individuals that have the disease as well as those who are caring for them, family members, friends, it's really important that we identify factors that can slow the onset and progression of the disease. So there are non-modifiable risk factors for Alzheimer's disease, and these include aging and genetics. These are things we can't change. However, there are other factors that we can change that can impact the progression of Alzheimer's disease, and one of those is diet. And there have been a number of studies that suggest that potential diets could help reduce Alzheimer's risk. But in humans, it's really hard to determine which factors, which macro or micronutrients in the diet are specifically responsible. And this is because our diets are so complex. Our genetics are so complex. So how individuals respond to different diets can differ. And then we also have gut microbiota, which can utilize and synthesize different macro and micronutrients. So in my lab, we use a much simpler system. We use the model organism C. elegans, and they have a super simple diet. They just eat E. coli, okay? So in this case, the E. coli is both their diet and their microbiome. There are many underlying factors that contribute to Alzheimer's disease. It's a multifactorial disorder. But one is the expression of toxic amyloid beta. And we have a C. elegans model that expresses A beta 1 to 42 in the body wall muscles. And now I can already tell what you're thinking. You're thinking, wait, muscles, not neurons? And the reason that there are also neuronal models that have A beta in the neurons, but the reason we use the muscle model, at least in this work, we are also working with the neuronal model, is because there is a really great readout for amyloid beta induced toxicity. And importantly, this A beta expression leads to AD-like pathological features in the cells that it's expressed in. It reduces ATP production, results in oxidative stress, causes defects in mitochondrial morphology, and these are all features that are observed in neurons of individuals that have Alzheimer's disease. But we have this very robust assay that allows us to identify factors that can affect this proteotoxicity. And so in this graph, these are the percent of animals that are moving, and this is the number of hours post-L4, fourth larval stage. And what you can see is that expression of amyloid beta causes robust paralysis in a time-dependent manner. And so our goal is to identify factors that alter this time to paralysis. So as I said, we were investigating the impact of genetic factors on this A-beta-induced paralysis, and in the process, we discovered that diet was having an effect. This is because we're using 2 different strains of E. coli. So we ended up looking actually at multiple strains of E. coli, and again, E. coli, just differing at a handful of different genes. And so OP50 is the E. coli strain that we normally grow the worms on. And HB101 and HT115, so OP50 is a B-type strain, and HB101 is a B-K12 hybrid, and HT115 is a K12. And you can see that growth on the HB101 or HT115 diets delayed the time to paralysis. We looked at amyloid beta in these animals, and here you can see the monomer, dimer, trimer, and higher order oligomers. And what we found was that the different diets did not impact amyloid beta levels. So they are protecting by a different mechanism. We discovered that A-beta animals grown on OP50, so this is a diet where they paralyze faster, had reduced levels of ATP compared to those raised on HB101. This is only observed in the A-beta animals, not in wild type. And while all A-beta animals, no matter what diet they were grown on, had higher levels of the reactive oxygen species superoxide, the HB101 strain, again, was protective and reduced reactive oxygen species. So at this point, we thought, okay, diet's having this protective effect. Why? And these strains actually differ in both macro and micronutrients. And we determined that the differences in glucose, fatty acids did not have an effect. And so we start to think about micronutrients, and there's this vitamin B12 reporter. It's an ACDH1 gene regulatory sequences driving GFP. And in animals that are B12 deficient, this promoter is turned, or this transgene is turned on, and they glow green. So you can see the animals that are raised on this diet that is detrimental, they were glowing green, whereas the animals that were raised on the protective diets, they were not. And so this suggests that the animals that were grown on OP50 were vitamin B12 deficient, and when we introduced B12 to them, you see the fluorescence went away. So we saw this striking pattern that in the animals that were B12 deficient, there was early paralysis, and the animals that were not B12 deficient, they had delayed paralysis. So we supplemented the animals, we supplemented the diet with vitamin B12, and we saw that vitamin B12 had a protective effect in animals that were fed this OP50 diet. And it did not have an additive effect for the animals that were already fed the HB101 diet that was vitamin B12 rich. And giving the animals more vitamin B12 didn't have any added extra benefit. I'll also note that, again, this is, as I showed you before, the animals fed OP50 have reduced ATP levels. And when these animals are supplemented with B12, this causes an increase in ATP levels. And again, the OP50 raised animals have higher superoxide, giving them vitamin B12 greatly reduced their reactive oxygen species. So at this point, we wanted to know, so there's 2 pathways or 2 enzymes that utilize B12. And I'm not going to talk about this part of the pathway because B12 is not acting through that part, and I only have a little bit of time. But we found, so B12 is required by methionine synthase to convert homocysteine to methionine. And then methionine is converted to S-adenosylmethionine, which is a major methyl donor in cells. And what we found is that mutations in these METR1 and SAMS1 enzymes eliminated that dietary shift. Okay, so now the HB101 diet was no longer protective. And so this suggests that this methionine SAMe pathway is required for B12 protection. We went further and then supplemented the animals with methionine, and we saw that that eliminated the dietary shift. And we added methionine to the METR1 mutants and saw that this partially rescued them. And this suggested that the B12 was activating this pathway specifically in worms and not just affecting bacterial metabolism. And we also saw this rescue, I didn't show you, but METR1 animals have greatly reduced ATP. And again, giving them methionine essentially bypasses this enzyme and raises their ATP levels. So I said S-adenosylmethionine is a major methyl donor in cells, so it can really affect the methylation, has epigenetic effects, but it can also impact the synthesis of phospholipids, and it's required for the production of phosphatidylcholine from phosphatidylethanolamin. And what we found is that supplementation with choline also eliminated the dietary shift. And so that suggested that B12 was having this impact by affecting phospholipid homeostasis. So I know that was pretty quick and a really kind of an overview. But again, in A-beta expressing worms, vitamin B12 increased energy output. I didn't show you this data, but it reduces mitochondrial fragmentation, and it decreases paralysis, but it does not do this by changing A-beta accumulation. I also didn't show this, but I'll bring it up, that when we supplement the animals only in adulthood, this is actually beneficial. So animals can be B12 deficient all throughout their development, and B12 still is fully beneficial when given to adults. Our results also show that the therapeutic potential is dependent on pre-existing B12 status, because the worms that were not B12 deficient did not get any additional benefit from being given more B12. And then we actually provided a mechanism. B12 protects against proteotoxic stress by acting as a cofactor for methionine synthase, and that increases methionine SAMe cycloactivity and affects phospholipid homeostasis. So, as mentioned at the beginning, I wrote an Alzheimer's disease supplement to the IMBRI grants, and this is work that I'm conducting with Jeff Kaplan in the Bioimaging Center and Chandra Kamanbetu in computer science. And we have a few main goals for this supplement grant. It's only a year, so it's really ambitious. Some of those goals are to continue to work on the story that I just described, and to really follow up on how SAMe-dependent phosphatidylcholine production is required for the B12 protective effects. And so we're looking at how the worms can sense membrane changes in membrane phospholipids and fluidity, and then how that impacts downstream pathways. We also have done RNA-seq, and we identified that this gene CBS1 is upregulated in the HB101 diet, and this suggests that there's a feedback mechanism to control the activity of this pathway. And so we're currently studying that feedback mechanism as well. We're looking into whether B12 has protective effects in other neurodegenerative models. And then finally, you probably didn't pick this up on this, but all of those paralysis assays require that someone count the number of moving worms every 2 hours for 18 hours. And so my graduate student, Andy, has spent many nights overnight in the lab counting worms, poking them, moving, paralyzed. And it's so low throughput. And so we are working to automate this paralysis assay because it's very robust, very black and white. And we're trying to do high content imaging using the new CD7 at the bioimaging facility, which would help us speed up the testing of various factors. And then we are going to be using a deep learning analysis to identify worms that are moving versus paralyzed, because they get a very, you can see the shape of their body really changes once they become paralyzed from this A-beta expression. And I would just like to thank the members of my lab, notably Kirsten Kervin. She was my first master's student. She was the one that identified that diet was having an effect on amyloid-beta toxicity. And my PhD student, Andy, who's going to be graduating soon, he did the vast majority of work for this project. And I'd like to thank the funding, UDRF, for an award, and also the Alzheimer's supplement to the Delaware Embry Grant. And that's it.